Hello, everyone. Good morning. Thank you all for joining. I'm going to go ahead and introduce the panel, but first, just to let you know, my name is Delara Motlagh. I am the Chief Operating Officer at FUJIFILM Cellular Dynamic. I will be moderating today's panel, and we're going to focus on some of the insights, reflections, and trends in the market that our panelists will share, particularly as we reflect on learnings that have happened in the industry that can help to support as developers are bringing the next wave of IPSC and pluripotent stem cell derived therapies to the market. So we have a fantastic panel here, and I will first allow them to introduce themselves, and then we'll get started. And just as a housekeeping, I will kind of look to the audience and make sure that we save some time for questions from the audience. But if you have a burning question before the end, please make a gesture, let me know, and I'll pause and make sure I invite you into the conversation so that we keep this very organic and interactive. Okay? So maybe with that,
Sure.
Seth?
Thanks, Delara. So good morning for you early risers. Seth Ettenberg, President and CEO of BlueRock Therapeutics. BlueRock Therapeutics is a pluripotent stem cell company. Our first asset is in a registrational phase 3 trial in Parkinson's disease that we've begun randomizing patients in. Our second clinical trial is in ophthalmology and inherited retinal diseases. We're in a collaboration with FCDI and OPSIS, bringing that product forward. And then our platform really is built to deliver cells into the CNS and into the eye. These are immune privileged, not immune desert, but immune privileged sites. And our hypothesis really is to find what cells are lost to the body in disease and aging, and then replace those cells with fully functional cells. Pleasure to be here and looking forward to the discussion.
Thank you.
Hi everyone, I'm Katy Spink. I'm the President and Chief Operating Officer and one of the managing partners at Dark Horse Consulting Group. Dark Horse Consulting Group helps clients with a range of strategic and operational development challenges across the cell and gene therapy industry. The reason I think I'm on this panel this morning is that pluripotent stem cells have a special place in my heart. I actually started my career at Geron Corporation, which was the first to bring a pluripotent stem cell product into the clinic. So look forward to having a conversation about this exciting technology.
Yep. Hello everyone, my name is Vanguel Trapkov. I lead our strategic options and assessment team for hematology, oncology, and cell therapy at Bristol Myers Squibb. And this is a team that focuses on portfolio decisions and really understanding trade-offs in mid to late development of the different options that we have in our internal pipeline. And actually, I share the interest in the special place of IPSCs in my heart as well. I'm a geneticist by training, and my Ph.D. was focused on hematopoietic stem cell generation. Failed miserably at it, but transdifferentiation, IPSC, and embryonic stem cells are the next thing that I'm very, very much looking forward to.
Excellent, thank you. So as you all can see, we have a fantastic panel with great experience and insights. So just to get things started, and Vanguel, maybe I'll start with you. If we reflect on some of the commercial therapies, and obviously your organization has spearheaded some of these now, what are some of the reflections that you have just on that pathway to commercialization?
Yeah, and thank you for that question. I would start by really reminding everyone the context of the first autologous cell therapies that came to the market, right? And this was the first time that the industry was facing with a genetically modified living product. But we knew that we had something transformational in our hands that could really transform patient outcomes. So from that perspective, there was a lot of challenges that we were figuring out, but the goal was really to bring that transformational efficacy to the market as soon as we can. So if we look at maybe some lessons learned and reflections on that, I would say there's three things that I would like to distill, right? Number one is, what worked well was the science, right? Number two would be the evidence generation and the partnership with regulatory bodies. And number three would be really the manufacturing perseverance, right? And from a science perspective, I think the CARs worked really well for a lot of patients. We really transformed patient lives through science. We moved from multiple rounds of chemotherapy for long years into a one-and-done paradigm with some of the outcomes happening in less than 30 days, right? I think for Emily Whitehead, that was 23 days. So really transformational efficacy there with, for example, taking some of our products like Breyanzi into consideration. Our transformed data demonstrated that, against transplant and chemo, like you were able to change outcomes from, survival in the low single digits to actually, 40+% of potential cures, right? So really transformative aspects. And I think some of the things that we saw in the industry as well was, payers started moving from a paradigm of usually they ask, why should we pay for something, right? To perhaps how do we pay for something, right? Also a problem, but a different one. If we look at some of the autologous cell therapies also, when they launched, they didn't even have DRG codes, but the ecosystem really put the effort and the work in getting these patients, the therapies to patients, and really institutions like Mass General Hospital and other centers of excellence really started fronting the financial risk, right, and still providing these patients with these CAR T therapies and then waiting for those codes to come up. On the evidence and partnerships, all of them were basically approved. All of the initial autologous were approved with single-arm trials, which really showed that manufacturers were able to demonstrate and partner with the FDA and regulators in order to really show the transformative benefit and the requirement for different type of regulatory pathways. And that also lessened the R&D strategic burden, right, when we were looking to develop into other indications, right? And right now, Breyanzi, for example, is in lymphoma has, the biggest range of indications that would have been possible if we had to do RCTs for everything, at least in the first generation. And then the CMC, right? From a manufacturing perspective, I think, our manufacturing teams and everyone's manufacturing teams have made, gargantuan efforts in improving the challenges for autologous cell therapies. We launched at supply constraints and, swapping processes, improving processes led to now being unconstrained, right, and being able to serve more patients. So all of these things actually contributed, even though that it was a challenging thing with a living product to actually improve, but we're still not there yet. We're still at a situation where looking at access, only 2 out of 10 eligible patients actually receive CAR T therapies. And this is something that we must solve together with the community, right? From a perspective of patients, physicians, payers, industry manufacturers, in order to improve, because that sustainability of the business model and ensuring proper access would be critical for the newer generation of therapies and for them to be successful.
Yeah. Really, really great insights, and I love the manufacturing persistence that you outlined, because I think persistence is a great word for some of the challenges that the industry has to manage through. But as you think about maybe even broader, and you gave great examples of just from the patient population and how you've kind of looked at that whole ecosystem to bring the product to market. Katy, I'm wondering from your perspective, as you look at other commercial products across the industry, what are some of the reflections that you have on their pathway?
Yeah, I would echo something that Vanguel said about transformational efficacy, and I would also frame that in the context of really, really tremendous unmet medical need. So if we look across the industry at the products within cell and gene that have been commercial successes, they've all been really addressing a very, very substantial unmet medical need where the patients have no other options and really terrible outcomes ahead of them. So that's true for the CAR T therapies. That's also true for Zolgensma, by far our most successful gene therapy. I think maybe as an industry, we have been optimistic about sort of still in our infancy, our ability to address slightly more incremental unmet needs, like for example in hemophilia, where patients are pretty well controlled by factor replacement, and what we're really looking to do is to avoid that need to infuse on a regular basis. But maybe that's not the level of transformation that we should be looking for with this class of therapies. I also would build a little bit on the manufacturing point and give, for example, the contrast between the launch of Kymriah and Yescarta. And we saw with Kymriah some challenges around their viability specification and their ability to deliver the product to patients in the early years that led to a little bit of a rockier start for their commercial launch. And I think that really speaks to one of the points Vanguel was making about needing to plan ahead for your CMC and really make sure that as you're going through development, you are pushing to develop a process that's going to be robust and scalable at your intended commercial scale.
Yeah, no, those are really great insights and across the different products that have already commercialized. You know, maybe Seth, as we now look at some of the challenges that the industry's faced, you know, can you reflect on what you've observed of some of the pathways that these products have taken?
As you talk about Yescarta and Kymriah having been part of the earliest of the Kymriah team without excuses, the speed to market versus the understanding and being first. Yeah. So I guess I start there with saying, this is new. Many of us are doing firsts. And when you're doing first and there is pressure by both capital efficiency and first to market, there are a lot of lessons to be learned and had. And I think you spoke of a really one that we all know well and was difficult from both a patient perspective and from a company perspective. And one that we should heed. In IPSC specifically, often I'll quote one of my colleagues at BlueRock that will say, when we talk about the newest, biggest obstacle we hadn't seen, they'll say, well, that's Monday. And that's just how the persistence that Vanguel spoke of is kind of like a little bit of what it takes with the talent of the team, keeping that team together. We are still an artisanal know-how field and can't underestimate how important then the talent is and the ecosystem that you're allowing them to both feel the pressure of the speed and the cost efficiency, but then get delivery. So the first lesson or common theme that I often think about is the efficiency of differentiation of the product. Start with all the lessons of drug development apply here. So often, and you spoke of it well around transformational versus incremental, we don't have that luxury in this field. It has to be unmet need, it has to be transformational, in the sense that understand your product profile and then understand exactly what that cell and cell type needs to be. And I'm still meeting with young companies that are still struggling with the production of the right cell type and not yet understanding their product profile, because the science is what's leading the birth of the company, which is right, but it has to be accompanied. So that lesson is true for us. Then it becomes efficiency of differentiation, characterization so you understand it, not to control it too quickly in the beginning. That lesson has to be learned and was learned in the early days of T-cell, of not overpromising the regulators. And then the next one would be the ability to scale and reproduce for the clinic. So it's one thing to make your batch for a preclinical model, and then even for lock it and do your potential GLP, it's a whole nother effort if you're going to have more than one batch for your clinical scale and reproducible characteristics, which mean get in conversations often and early with your regulator, whether that be the FDA or the EMA. And those have been, for us, incredibly important. A story that doesn't often get told for BlueRock is that its first IND was on a 2-year clinical hold when I first got to the company 5.5 years ago. A lot of that has to do with the firsts. The FDA hadn't yet dealt with a product like this, in the brain, and there was a ton of education going back and forth for BlueRock and the FDA, and that was a partnership. And I think that's the last place I'll leave it with. On the stage, I'm partnering with at least 2 of these organizations. Vanguel, we'll get to it, but we haven't yet partnered. And it's really important in this field. I think you can be really, really good at some of it, but being really good at all of it, we have been well capitalized, and we have now stood up for about 9 years almost, 8.5, 9 years. And we like to think of ourselves to end, but we still need quite a bit of help in device. We still need quite a help a bit in automation of manufacturing as we go to tremendous scale or think about tremendous scale. So I think our field in its infancy still needs to think about strategic relationships that might not be ones that you would enter into if you were a biologic, so to speak.
Yeah, no, I think that the partnerships is one of the things in this industry that I find heartwarming, right? Because it's, you know, on one hand, there's a lot of secrecy, or at least there has been. I think we're moving a little bit away from that. And but at the other end, you can't really go at it alone. And so that all the different partnerships are so important. But the regulatory piece, I think, is also really key. And I appreciate you sharing that challenge that you took on when you first, you know, took the helm at BlueRock. You know, Katy, as we look at the regulatory environment now, I mean, we know that there's been a lot of ambiguity. These are novel therapies, as Seth very nicely articulated. But we're also in an era of uncertainty where it comes to, you know, the political and regulatory climate. So can you highlight maybe what are you observing as far as these trends and what it means for us and implications?
Yeah, I mean, taking a long view around the regulatory environment, so comparing today to, you know, almost 20 years ago now when Geron filed its IND, I would say there's actually a lot more flexibility built into the regulatory environment than there was back then. So I think, Seth, you talked about some of the being first and the unknown risks, right? And so the Geron IND was over 20,000 pages. It had thousands and thousands of animals out to really long time frames. And the primary concern there at the time, which I think we all shared, was the risk of teratomas from these cells, right? And really, how do you get your head around what could be a rare event that would take a long time to develop in those studies? I think as we've gotten decades of experience with tens, probably even hundreds of patients now, and not seen those types of safety risks, I think the FDA takes a more balanced view, and we're seeing a lot of flexibility around things where there used to be none. So around things like design of the IND-enabling tumorigenicity study and what the length of that has to be and what spiking groups you need to include, for example, depending on the MOA of your therapy, depending on how good your analytical package is around residual pluripotent cells and other concerning impurities, et cetera. Now, the level of interaction, however, I would say has been also very different in a negative way for the field. So, you know, Seth, you mentioned having that back and forth with the FDA. We had that at Geron as well. These days, I would say there's less opportunity to just call up your reviewer once a week and ask a question that we had back then. That said, that's partly because there are fewer questions that really need to be asked of the regulator because the industry has defined answers to those. Also, I would say compared to maybe about 2021, when you could really see that the agency was very stretched with all of the applications and not enough staff to review them all. I would say we've gotten back to a little more normalcy in terms of the interactions with the agency, and we've also had new programs developed that have created more opportunities to work with the regulators early and often. So pre-IND already existed, but now it's been formalized as Interact. It's actually a PDUFA meeting. We have Type D meetings. We have the CDRP program. We have RMAT. So there's a lot of opportunities that have been created for more formal interactions with the regulators. The other thing that I would say has changed and has influenced the regulatory environment is there are a lot more tools and technologies available to developers these days.
Yeah.
So back 20 years ago at Geron, we were calling up our vendors, begging them to make cytokines GMP for us, and they wouldn't because, you know, we were the only ones. So you had to buy a large lot of research grade, do all of the testing yourself to de-risk it. These days you have your choice of vendors who offer, you know, high quality reagents and materials. You don't have to use Matrigel, you can use recombinant laminin. And there's also great tools and technologies for analytical characterization of the cells that didn't exist. And so that's a good thing for the industry. That also means that regulators expect you to do the best you can for your therapy. So it means they've raised the bar on raw materials. They've raised the bar on their expectations for your analytical package, appropriately so.
If I could, go ahead.
Sorry,
yeah,
please.
go for it.
If I can build on that point on the regulatory environment, I think, what has remained constant, and I think that was mentioned also in some of the panels yesterday, is, the partnership with FDA, right? And the FDA regulatory bodies really working with manufacturers to understand the living products and what we're dealing with has been consistent, right? I think I 100% agree on the interactions. I think the early autologous cell therapies did benefit with very frequent interactions with regulatory bodies. I think now, like looking just at the ARM latest reports of clinical trials, we have close to 830 phase 1 cell and gene therapy trials, right? So I think as a trend, maybe those, even though some of the tools and different kind of ways to look at cell and gene therapies have evolved from a regulatory perspective, the frequency of interaction might be a little bit less so. It might be also that, we see less RMATs as well, right? As part of just the general wave of INDs that is coming in. But I think also linking to something that Seth said about really understanding the product, I think that's where, BMS has also put a lot of effort in demonstrating that, the kind of the regulatory and the approval of your product is not where it ends in terms of data generation. In order to truly understand it, not only from an efficacy and safety perspective, but from a scalability, manufacturing, and which unmet needs in the COE setting, in the community setting, you'll be able to address. You need to constantly have a very, persistent focus on generating data, generating consortiums, and creating consortiums of talking about this data. Really driving the ecosystem's knowledge of this living product in order to really characterize it from all different perspectives. So I think that's important, and that has also led to some of the tailwinds, such as the data-driven removal of the REMS program, which BMS actually put a lot of effort to generate data, and together with ARM and their leadership was able to really demonstrate that certain regulations make sense, others maybe can be alleviated, right, in order to then increase access for patients and really make these tremendous therapies accessible to more patients, right? So working with the regulators has remained the same, ensuring that the data generation and really informing about what your product is, what unmet needs it would address, and then what challenges it also creates, right, is very important. But you cannot do that without the data generation and really the understanding for yourself what your product is, and then driving the ecosystem's understanding.
Yeah, it's perfect. You picked up exactly where I was going to jump off on the RMAT conversation, because so we filed RMAT and received RMAT designation. It's been incredibly helpful. It moved us from phase 1 to phase 3. I think what we often forget is we think mostly what the FDA is concerned about is safety, feasibility, and efficacy, and that's why you get RMAT. Actually, for us, a large part of that conversation was our first comparability study, because we went from phase 1 into our next phase, phase 3, with a comparability study, and to go to commercial, we're going to need another comparability study. And both our plan and the actual data we provided them with and the path to launch was equally important to them, and that CMC package was equally important to them, as was the safety and the efficacy data that they were seeing early signs of. And so you can't underestimate the value of that data. And it actually puts a lot of pressure on our field in terms of cost, because building that data set means you have a ton of retains and a ton of vials that you're not putting towards patients, and that becomes really costly. So thinking ahead on, again, back to my comment on manufacturing scale and efficiency, that you have to think that through than just getting through your first 10 patients, which when you're first starting out is very hard to do.
Yeah, those are, it's, when you reflect on some of these learnings from a regulatory perspective, I mean, the industry is evolving, but the regulatory landscape is evolving at a very fast clip. And just as we solve some issues, other ones pop up, right? And so it's important to keep a pulse, and the data generation and the analytics are so key, particularly with these very novel therapies. Seth, I'm wondering if you can also just you know, as we think about some of these learnings, and you've already highlighted some that are specific to the pluripotent and the IPSC, but what other kind of unique considerations in the development of these therapies do you see?
So maybe it's starting to sound like a broken record, but I guess because of we've tech transferred into our commercial manufacturing facility and are now producing commercial grade cells from Berkeley, California, dopaminergic neurons. I would guess the only persons on the world doing that right now at that scale. And what I would caution and/or want, and we're seeing the correction already in the field. Many of us went out and bought our own manufacturing teams and manufacturing facilities. That capital's not there right now for most companies. I think thinking through how you stay in touch with and next to your manufacturing is really important. You can't simply let go and let someone else, even if it's a T cell and others have made T cell, even if it's a cardiac cell and others have made, or a dopaminergic neuron and others have made, you have to stay incredibly close to the work there. So the first bit is just around the manufacturing and the scaling. And I hope for our industry that we start to get to more of a standardization here. We saw it happen in biologics, where we went to DMFs, we went to clones, we were able to actually share clones and start to insert, and now we're inserting DNA into standardized clones. I don't think we're there yet in our own field, but I imagine a day where we can file a DMF and all of the critical early reagents aren't as thought of as much anymore. And I think for us that would be incredibly powerful for the field. And then we've been talking a little bit about it, but the interaction and the complexities of our regulatory paths, which are to be defined, and we're defining them, and our payer path. And both of those pieces are now incredibly ambiguous for our field, and it is upon us to define them by working closely with the regulators on that. The ambiguity and the change that team is going through right now is likely going to create time and space for us that we weren't ready for. And then the payer side, I think I would tell you that from the early days in T-cell, autologous T-cell, I've been really disappointed in the innovation we have been unable to apply to the payer situation. Yes, we got a new DRG, but we still haven't really figured out how to go from a one-time therapy payment system and make that affordable and usable and greater market access for patients instead of the one-time payments. So I know that that's difficult, and many have tried, so it's not to say that people aren't trying, but I think that also is a potential opener for our field to figure out a way that works to get more patients, as you said, 2 out of 10. That's just not, when it's life-saving therapy, that's not acceptable. So I think those are the pieces that I'm most focused on: standardization, regulatory path, payer path, and manufacturing.
Yeah.
So one thing that I would add is when we think about the pluripotent stem cell space, it's really pretty unique within at least cell therapy in that we have the ability to employ a model that is very analogous to traditional biologics, where we have a master cell bank and a working cell bank, and we're using that master cell bank for the entire product life cycle. Mm-hmm. Because of the nature of small, innovative, early-stage biotech companies that are often developing these products early in their life, there can be a necessity to focus on kind of the next milestone and the next milestone. And one danger that I think exists for pluripotent stem cell companies is not putting enough investment into making sure that their master cell bank is going to be suitable for their entire product life cycle. So a few potential pitfalls there include donor eligibility and the fact that requirements are different globally. So really thinking about whether the cell line that you're using will meet global donor eligibility requirements. Thinking about the standard by which it's manufactured and making sure that that will supply it. And also looking at looking deeply at it for any genetic mutations that may exist that could potentially cause safety concerns. And then finally, this isn't only about the master cell bank, but I think thinking about where do you ultimately have to be at commercial scale and making sure that you're planning ahead for how you're going to get there, even though you're not going to be there in phase one, right? So, for example, making sure that your manufacturing process is going to be robust out to the passage numbers that you're ultimately going to need to make lots of that size.
Yeah.
I think those are fairly unique considerations to the pluripotent stem cell space that sometimes are not obvious to individuals who've been, elsewhere within cell and gene therapy, and also, very understandably are just focused on, let's get in the clinic. We need to get in the clinic to raise the next round, right?
Katie, I wonder so it's really good that you bring up both the genetic stability and mutational landscape at the start. I think the other piece that people get freaked out about are there are genetic changes. If you take 2 skin cells out of your body right now, they will have genetic differences in them. And the important piece is to understand, as the FDA and we all increase the bar on our ability to characterize these cells, is creating a rational path towards that scale. In that and setting the expectations and verbally and willingly talking with the FDA around those expectations is really important.
Yeah, and we actually see quite a bit of regulatory flexibility around that. The FDA is looking for you to define the path of how you're going to look at genetic stability and to justify it, and we've seen a number of different approaches be successful. And I would caution developers, it can be tempting to just look really, really deeply for any, genetic change, but if you do that, you are going to find things that you don't know what they mean, right? So instead, doing a prospective plan for what are the things that I may be concerned about, and designing a rational genetic stability program to fulfill that prospective plan, I think is what we typically advise our clients to do.
Yeah, that's really, I think, a big one, right? That people get very concerned about, because you're right. I mean, how do you process data of things that you don't know? Because we're all very different. Our genomes will reflect that, and there will be fluctuations and differences, and even mutations that may not mean anything. But contextualizing that, I think, is so key. So I'm really, really glad that you've addressed that. Let me now, as we think about, right, just some of these elements, particularly some of the unique elements for the pluripotent, Katy, as you think about the investors, because we've talked about the need, right, and Seth said very nicely that, we really need to analyze and have these analytics for our products, but they're costly, and we know that from an investment perspective, there are challenges that the industry is facing.
Yeah.
So from your vantage point, how are investors looking at these opportunities, and how are they assessing them? What are they looking for?
Yeah, so I would say I'm seeing a dichotomy between your more traditional investors, venture capital, and big pharma. So starting with venture capital investors, I would say there is a fair amount of skepticism in the marketplace right now. I think there's a tendency to say, it's been 20 plus years, we haven't had a success story yet. Maybe the timelines are not compatible with their typical timelines. I think one of the things about pluripotent stem cells is the upfront investment in both time and money is greater. It will take longer to get into the clinic with a pluripotent stem cell product than it will with a autologous T cell, for example. The downstream payoff, though, is great. And so what we're seeing is that there is a surprising amount of investment from big pharma in this space. So
despite what you may hear in Fierce.
Yeah, I mean, so Bayer, Vertex, Roche Genentech, Astellas, Novo. And I think the reason for that is big pharma is really optimally positioned to understand the potential upside of these therapies, which comes back to what I was talking about earlier with them really being able to mimic the model that big pharma is more used to and knows how to commercialize, which is scalable, off the shelf. products. Now, I do think if BlueRock or Vertex is successful in their pivotal trial, as we've seen time and again with investors, there's sort of a lemming effect there, right? Where, or maybe lemming's not the right analogy since this is a positive thing, but there's a follow the leader, like FOMO type effect, right? Where I think as soon as we have our first real success story, we're going to see a lot more interest in the space from investors. But for the time being, I would say there is a fair amount of skepticism in the venture community about pluripotent stem cell therapies.
Interesting. Anything to add from Seth or from Vanguel regarding the investor community?
Yeah, I mean, I agree with the skepticism. I'm reminded in the audience sitting here of every time BlueRock has success, our competitors come and say, thank you, that helped our next round of raise, which I think actually is important for the field. The common diseases that we're entering into are millions of patients. We owe it to them to get this right, and as in many times over, in the T-cell field, we learned with many coming in, it actually offered patients the greatest benefit. And so I think we are seeing already a bit of that wind, as long as capital is at the cost it is. And ARM actually put out a really nice slide deck, Reasons to Believe, that have 2 or 3 data sets that I'm finding very helpful with investors, which are, yes, the upfront investment is greater, but the likelihood of success into phase 1 and the data from phase 1 is higher than in a small molecule or a biologic. So you can run 3 or 4 biologics programs for the cost of my one, but I'm much more likely. And that comes down to a little bit of what I often tell others to pay attention to, which is, do you understand engraftment? Because we're transplanting a cell that's not endogenous. Do you understand the biological activity of your cell? Because then the preclinical model to me means a little bit less. So we have a cell type, an oligodendrocyte, that myelinates neurons. If you understand that function and then think about which diseases to apply it in, you become to be able to go across multiple indications. And so that step is a little bit more robust for us in the field, because that function is what's required, and then understanding the disease and as we talked earlier, the TPP that you have to have for moving into that indication. So as we get more and more examples demonstrating that, and across diabetes, the epilepsy data set right now is incredible and stunning. Parkinson's data set across multiple labs is now becoming, like we remember in the CD19 days, of the same biology, but different labs making different products, showing us sort of the same data. So that, I agree with you, that as we validate through, the last risk point is really around commercial viability and launch, which is to be seen, and that's going to take a long time.
Yeah.
It is, and if we reflect on some of the other therapies and the timeline that they've taken and what has happened when they have commercialized, I think these pluripotent stem cells are on that same journey. But Seth, as you pointed out, what I also find with cell and gene overall, but I think particularly with these specific cell types, is that probability of success, right? And knowing that these can be truly, going after some of these transformative and regenerative issues versus, some of the others gives a lot of hope. But if we think about now, the IPSC therapies overall, and Vanguel, I would be curious your thoughts, right? As you think about some of the challenges that we articulated overall for cell therapy and some of the things that we've been discussing on the panel on, this path to commercialization and lessons learned, what are some of the things that you think that IPSCs are, able to address that are challenges that other cell therapies continue to face?
Yes, and I think it's important to focus on what the problem we're trying to really solve for is, and I think for us that's democratizing cell therapies, right? And when I think about the democratization of cell therapies, it's I'd say 3 elements. Number 1 is really improving the current access, like as we discussed, the 2 out of 10 paradigm is something that really needs to be addressed, but also where I think the newer technologies can also come and really address some of the unmet needs are I come from a portfolio decisions group, and I think the beautiful thing about cell and gene therapies is that now we suddenly have therapies that can address a variety of very, very different diseases, right? If we take autoimmune, for example, with CD19, we can potentially transform the lives of patients with all different B-cell related autoimmune disorders, right? By that, then from a portfolio decisions where we look at the total value, the opportunities, what makes strategic sense to expand in, the bar is very high with autologous cell therapies, right? So I think being able to have a paradigm that allows us to expand into additional indications, right, that might not be sustainable currently with current technologies will be critical. And then one other piece of democratizing cell therapies is also, and we don't speak about it enough, I would say, is we want to also expand the country footprint, right? Cell and gene therapies can now be a paradigm that is only, in EU5 and a couple of select countries. And I think that, with the newer technologies, with, focusing maybe on the cost of goods, addressing some of the paradigms, learning from 8 years of, autologous developments, and really developing the products to address that unmet need, be it in the community setting, in, from a access and payer setting, would really help us also expand that country footprint, which I find critical as well.
No, those are really great insights. And Seth, as you think about some of the points that you made around scalability and some of the things that kind of keep you up at night, how do you see this in iPSCs in particular as you look at their potential and maybe a path that you may take that's different with a pluripotent stem cell versus a conventional stem cell?
Yeah, I think Katy said it really nicely in the sense that we start to take lessons we learn from the biologics in a standard master cell bank into, and so the lessons that we saw there were around standardization, almost commoditization of those processes that went across the field. And you can go to different CDMOs, but you're basically getting the same services. And I think I look forward to when that's happening for us. And much of that, as I imagine, and we're obviously investing into, is automation, into some of the sort of traditional steps that you have to go from a small cell adherent into your then larger scale pluripotent cells, then into your differentiation process. The differentiation process will be unique and different to everybody, but those earlier steps are going to be very common steps. And so looking for solutions and automation around some of those common steps, I think are going to be incredibly helpful to the field. And then standardization, as each of us creates, you talked about the tumorigenicity and the distribution study, and as each of us creates that data set, we're starting to reproduce data across a very select group of cell types that I hope also creates an understanding with ourselves and the regulators that we can start to democratize, I like that word, more and more. And we've already seen that. We're seeing that the time frame the FDA is expecting to get shorter. But the burden we all have to stare straight in the face for is the cost and the time frame. That is just, it's not yet viable to continue to do that across as many cell types as this technology allows us to do. And BlueRock's gone through 2 iterations now of focusing down because of that. We had many, many more cell types in our quivers, in our sheath, because of all the cells that we could make, but the reality was the cost of that is unbearable. And so I think that's an area where I'm very interested in partnering and looking to others to help bring solutions as well.
I'm sorry, Katy, go ahead.
No, and I was just going to say, and I think you alluded to this with your myelinating cell. I think because it's very challenging to figure out how to differentiate a pluripotent cell that could be any cell type of the human body, reproducibly, scalably, reliably, cost-effectively into exactly the cell you want. There's a lot of benefit to be had from thinking about expanding indications of the cell types that you've invested in making that process, rather than trying to do, 6, 8 different cell types, right? And the myelinating cell type is a great example of that, where there's a pretty wide range of indications that you can go
The one I'm
after
most
with
excited
that.
about is the microglial cell, which for me, it's that same idea that the T cell field has really benefited from, that they can keep using that chassis to do different indications and spaces.
Yes.
That's the microglial cell for me, has that same sort of appetite for it.
Yeah.
And that's when you talk about the investment and the time that it takes, particularly with a lot of the upfront work, I think these are great insights. What other, pathways or ways can you accelerate? Because obviously, getting not only into clinic, but to be able to commercialize faster with these therapies will solve a lot of the things that we just discussed. So aside from kind of taking a process and finding additional indications that you can leverage, what are some other ideas to accelerate that pathway to commercialization?
Yeah, I would say being really thoughtful about what pieces you need to own that need to underlie the proprietary nature of your product and which ones you can take off the shelf, right? So the answer is not going to be the same for everyone, but for example, there are now very good cell lines out there that you can license. Do you really need to own your cell line, or can you own your manufacturing process? Same thing with delivery devices. Is there a delivery device that's out there that you can repurpose to deliver your cells, or do you have to develop a novel one? And sometimes the answer to those questions is going to be, yes, I have to. There is no existing delivery device that can do exactly what I need it to do. But if there is, leverage that. You don't need to, it's hard enough developing the cell type that you're developing and showing that it's safe and efficacious. You don't need to be needing to pave the roads left and right of the one you're on as well.
Yeah, I also think a lot about the accelerated pathways available to us that are already existing, and maybe not as particular to Parkinson's per se. It's still a growing field in biomarker, but thinking through as you enter, is there a biomarker, or can you produce a biomarker from your cell type that allows you to demonstrate that once you have engraftment and once you have the functionality out of that cell, can you now say, well, then this will predict my efficacy for the regulators and for the payers? I think is a really important one. We're following F-dopa PET because it's the function of the cells once they're there. And you can imagine that that signal can one day be used as a first goer. We're not really going to be able to use that, but I can imagine that the field in Parkinson's will benefit greatly. And in regenerative medicine, we're doing things that no one in the payers or the regulators have ever thought about or thought possible. When you bring back a function of a cell type, we're not surviving a cell and looking for markers of that. We're actually bringing a brand new young cell to a function that did exist in the body but no longer. So this is exactly as I think in our ophthalmology clinic, where yes, many of the gene therapies are keeping those cells alive, but we're actually adding cells that didn't exist there, are different from the patients, and so there's all kinds of a marker status that you can imagine. I'm really excited about moving into Alzheimer's in the future, and here I think the burden that I start to get stuck with is it's such a large patient population. Imagining surgery for all those patients to implant cells becomes difficult, and so we're looking for technologies that would allow us to maybe skip some of those needs to go into a standardized OR room, but into a more simpler procedure that allows us to get the cells to the right place at the right time.
Yeah, I would also say taking some sort of baby steps in your proof of concept, right? So I think a good example of this is the Vertex diabetes program, right? We, as an industry, struggled for literally decades to develop immunoisolation devices without success. So in their program that's in a pivotal trial now, they have adopted the methodology that's been used for many decades with primary cadaveric islets of the Edmonton protocol, and just proving the function of the cells themselves without having to think about, novel delivery route, novel way of protecting from the immune system, I think is a great way to demonstrate the potential. It limits you to a smaller population of more brittle diabetics, but then it allows you to build out from there once you've established those initial steps, rather than taking risk on multiple fronts at once.
Yeah, no, those are really great examples. I'm just going to pause and look to the audience to see if there's any burning questions or Any questions that you may have? Yes?
Hi, thank you for the conversation. It's excellent. I'm wondering about actually scaling for commercial populations. Where are we now? What needs to be done? How close are we to making that feasibility? You brought up type 1 diabetes, and that's a good example. Parkinson's is also quite a large indication. So how are we going to actually scale the iPSC cells to reach these populations?
You want me or you?
You're the one who's doing it.
I'm not doing it. Okay. So it's a great question, and one that we obviously are right now wrestling with and working on, and I can tell you how we're thinking about it. So first, break Parkinson's down from the 10 million. We're not talking about 10 million patients. We're talking about actually moderate Parkinson's, which is now actually the bulk of Parkinson's. And then it's those that are going to be in the stage of their disease where their drugs are no longer controlling their movement and they're having a certain number of hours per day where they optimize drug, but they still are not getting benefit from their drug. So now that whittles down the population yet again. And then often, as we saw with the T-cell therapies, I think also setting expectations for launch is really important. That we're talking about a neurosurgery of the building of the capacity and the capability, which if you go back to cardiac, for stents and for surgeries that didn't exist before we had those surgeries, it took time to get doctors trained and the ORs trained. Now, luckily for us, the surgery that we're doing is actually quite simple for a neurosurgeon, although I know we're putting needles in brains, so I don't want to underestimate it. It's quite simple and quite common to what they do. But it's still needing OR time and still needing surgeons. And so you can start to imagine the kind of launch filter that we're looking at is one that's addressable and one where today we are capable of that scale. Where it goes, I would say to you, 5 to 10 years after launch, that's more of what keeps me up at night for, with success and with a good launch ramp that allows for those capabilities to be built across the major, and you said it really nicely, Vanguel, we're really focused on the major economies that can deal with this. That's the piece that where we're not quite there, and that's where automation is really coming and stepping in, and we're becoming dependent on it. We won't be there all fully automated through, but we have a really beautiful facility in Berkeley, California, and a team that has already been 2 years into the tech transfer, which is about what it takes to get that stood up. And so I feel quite confident for the launch, but beyond that, with success, is one that we have to work towards.
Excellent. Thank you. It's an important question, and thank you for going through how you're looking at it and some of the specificity, which I think is really key. And I think a good problem that we will all have in the future is that when these are wildly successful is that additional scale that comes beyond. Any other questions from the audience? Okay, so if I come back to the panel, I know we've already shared some guidance and feedback and insights, but I'd like to ask each of you, if there's a developer for iPSCs in the audience or beyond, what advice would you give them just based on your reflections in this discussion? Maybe Vanguel, I'll start with you. What advice and guidance would you provide?
Well, if I can boil it up to one thing, I'll echo some of the things said today, as well as, I think yesterday Christine Yarema put it very nicely. When, as you're developing a technology that is new, and again, it's a living product, you really need to start with the end in mind, right? I think that's the words that you used. And that can be kind of split into the commercial end in mind, and I think Seth also mentioned the TPP, and what are the unmet needs that you would be addressing? And that is the challenge, I think, with newer technologies, is that, the unmet needs evolve, and how that would change in the next 8 or more years is an evolving question. But then also, when we're looking at the TPP, I think, a friend of mine, George Smith from Destin Therapeutics, taught me 8 years ago that, when you were talking about cell and gene therapies, phase 1 is when you plan for your commercial success, right? And when you go to the registrational study, that's when you plan for registrational approval, right? But when you hit the market, you need to have a really good data package, right? And that would probably still remain to be the case in the future as well. And we know that, be it a single arm trial or a RCT would not be enough to fully characterize a living product, right? So generating that data, ensuring that you have as much data as possible, thinking with the end in mind, is just critical for success.
Excellent, thank you. Katy?
Yeah, I mean, I think my number one would be the TPP, but since Vanguel covered that beautifully, I would say second on the list would be really planning for your CMC development life cycle. And I touched on a couple of the key points, and actually Seth touched on one as well that I would mention here, which is you don't need to have your commercial process from day one. You probably shouldn't. The amount of time and money that it would take to develop that before you ever even get into the clinic would be astronomical. You'd never get funded, you'd never get in the clinic. But you need to have the vision of what that process is going to look like and the roadmap of how you're going to get there through development and what changes you're going to make when to avoid painting yourself into a corner from a comparability standpoint. And the other thing I would say is to recognize that the field is going to evolve rapidly during that development, and so you want to position yourself as best as possible to take advantage of that. And Seth, you used one of my favorite words earlier, which is retains.
It's not my favorite word.
And yeah, it can be costly, but really thinking about, analytical technologies have come so far over the past couple of decades. And one of the things that we saw at Geron and subsequently at Asterias was that we were really well served by the fact that we have been taking retains, not just of the final product, but throughout the differentiation process. And that allowed us then, when we got to the need to put together a comparability package, that allowed us to show not only that our cells at the end were similar to what they had been previously, but that they were following the same developmental lineage along the way, which I think gave us a much more compelling argument there. So it can be really painful. There's obvious trade-offs there, but making sure that you're being really thoughtful about your retain strategy. And then the last thing that I'll say, and it's become a bit of a generic thing in the field, but I'll say it anyway because it's so important, thinking really early about your potency matrix and your potency strategy. It is really challenging for cell and gene broadly. It is even more challenging for most pluripotent stem cell derived therapies because they're in this, many of them are in this more regenerative space where there's a very complex and multifaceted mechanism of action involved.
And you're putting a progenitor in, you're not putting the final product in. So the complexity of that, and I like that you use the word matrix. Nobody says assay anymore.
Yeah.
We just simply say because it's going to be multiples. As I think through, just to cause a little friction between you and I on this, I think it's important that we don't overbuild in the beginning and take some of those lessons, and that we pay really close attention to something you said in the earlier part around the ability to transform. And I think that as many come into field and start to crowd around certain objects, I think it's a time to stop and look. There is so much low-hanging fruit left in creation of new cells that there are places to go, and I want to encourage developers, and especially some of the young developers I meet in Cambridge where I'm housed, to go after some of that and not to be daunted by the cost or the length of time. And I think it's truly, I'm watching a really exciting field develop around us. And so I also want to throw a little bit of, yes, this is costly and long and be really careful and scalable and reproducible, but it's doable. I think that's one of the things that I come to this field from the T-cell field in autologous. That brought me here because of how difficult that reproducibility was. And I can tell you now at scale in commercial facility, this can be a reproducible bit. And so I also want to encourage those that are thinking of brand new modalities that this is the time.
Absolutely.
Excellent. some great guidance and feedback and a lot of opportunity in the field. So I'd like to first thank all of you for spending your morning with us. And I would really like to thank the panel for sharing all of your great feedback and insights and experience as we navigate, you know, some difficult waters, but I think fruitful ones, and some really great guidance. I'll leave you with, you know, I think Seth said it at the beginning of just the partnerships in this space. So you're not in it alone. There's a lot of patients that are counting on us to get it right, particularly for these very transformative therapies, and having these strong partnerships in the industry, because you don't have to do everything on your own. So thank you again for the time, and thank you to the panel.
Thank you,
We'll
Delara.
close here. Thank you.